Evening. We'll do it one more time. Good evening. Welcome to the Shoals Marine Laboratories weekly seminar series. I'm Sarah Morris, the John M. Kingsbury Executive Director of the Shoals Marine Lab, and I want to thank everyone for joining us tonight for our 11th Rock Talk of this 2026 season. For anyone who is new to the lab, and for those of you who are joining us online, the Shoals Marine Lab is the oldest and largest marine lab dedicated to undergraduate training, both in terms of education and in terms of research. We're jointly operated by Cornell University and the University of New Hampshire. Our seminars are affectionately called Rock Talks because back in the day, seminars, lectures, what have you, actually happened literally on the rocks. On a day like today, when we had the deluge just before dinner, it's a really good thing that we've moved inside instead of being outside on the rocks. And this is a great opportunity for everyone in our community to come together and learn things about science that is happening in this area related to the marine environment, to general environment, to climate change, any topics that might be interesting to a wide range of audience. In addition to our guests joining us online this evening, we do have 3 new classes started on Monday. We have sharks, skates, and rays. We have marine vertebrates of the Gulf of Maine. And this week, we also have a visiting class from Anne Arundel Community College in Maryland. I love the enthusiasm. In addition to the classes that are here right now, we do have our 10-week undergraduate researchers, our Shoals undergraduate research group, Surges, who are getting close to their own symposium. For anybody who's interested, watch our newsletter to see when they will be presenting. We're excited to have everyone both online and in person today joining us for this seminar. The format of the seminar is that we will have a 45-minute talk followed by approximately 15 minutes of Q&A. For any friends who are online, please use the Q&A button at the bottom of your Zoom screen to ask questions, and we will ask them so that they can be heard. For those of you that are in the audience, I will be coming around with a microphone so that the people online can hear the questions in addition to the answers. Tonight's speaker is Dr. Stephanie Hurd. Steph is a postdoctoral researcher at the University of Massachusetts in Boston, studying predator-prey dynamics between brown bears and salmon in Alaska, the collective behavior of these migrating salmon, and how predation, environmental conditions, and social learning influence individual and group decisions. Previously, she was also a postdoctoral researcher in the Virus Plus lab at UMass Boston. Her research investigated connections between climate change and anthropogenic drivers in gull colonies and avian influenza transmission in Alaska. Steph earned her PhD in ecology and environmental science from the University of Maine, Orono, where she studied the cascading effects of forest management practices on wildlife communities and tick-borne pathogen transmission. She graduated with a BS in biology from Quinnipiac University in 2016. Prior to graduate school, Steph worked on various research projects in the Northwestern Hawaiian Islands, Minnesota, and Utah. Overall, Steph continues to have a strong interest in community ecology, predator-prey dynamics, trophic cascades, wildlife conservation, and disease ecology. She is fascinated by the cascading effects management practices have on ecological systems. We're also thrilled to say that she is a key member of the Gulls of Appledore banding team. This evening, Steph will be presenting the effects of forest management practices across spatial scales on tick densities, wildlife communities, and tick-borne pathogen transmission. Please join me in welcoming Steph Hurd.
I was stuck out. Those goes right about there. So it's part of the Papahana Marine National Monument for the Northwestern Hawaiian Islands. I was on a little bit of dance called Surrey Atoll, and it put that into perspective what it was like out there. What has been maybe castaway on Hank. Yep. All right. So that was neat. We were out there disconnected from the outside world. Our water collected rainwater, no internet, just stars and shadows at night. That was about it. So I went a little feral, grew a little bit of beard, not so. But while I was out there, I got to study things in albatross, Hawaiian seals, got to look at different species, see how all of these animals coexist on this little mile long. After taking my beard, I then went to the cold tundra of Minnesota for 3 months in the winter, collecting yellow snow, which we normally try to avoid, but I wanted it. It was cold because I was looking at moose and doing a nutritional study to look at their health in the winter. After the cold, I went to the hot, and I went to Utah. And there I got to study wild horse and burro. I did a behavioral study, and I looked at the different herons and how those interaction with one another and how they're born, vision, fusion. And I got to look at some stuff that was fun. So, how did I get to tips? One thing I'll ask, feel comfortable, who here has either had Lyme disease or no someone who has done it? For me, I grew up around ticks. I grew up in Connecticut. My childhood friend had chronic Lyme. That really put it on the map for me. But really, I got into tick research because of the fashion. So, when you collect ticks, one really common way of doing so is drag sampling. I like to call it my ghost hunt stuff. So, the fashion really brought me in. I really loved it. But more than the fashion, it's messy. So why I told you all that history of the research that I've done, I've been all over the place. I've looked at different animals and all these little complex interactions, and that actually lends itself really well studying because they have an extremely complex life cycle, and it's really embedded with important. So the tick I'm talking about is going to be the one that transmits the bacteria. Oh, so that's a little history. So Ixodidae damni or Ixodidae scapularis now is known as a deer tick, but is a nerd. The very correct term is actually fragmentation. So that is the tick that transmits bacterium that causes Lyme disease, and pictured here is the life cycle of that black-legged tick. So to walk you through it, start off where there are eggs that are laid, and they hatch into larvae, and in this larval stage, that's the first chance for the tick to take a blood meal from a vertebrate host. This is important. Ticks are not born carrying Lyme. The only way they cure that bacteria that they pass on when they bite off their animals is by feeding on an already infected host. So it's a little bit of chicken and egg. It infects the animal infects the tick. No. After the larval stage, the tick will overwinter, and then it'll molt again. The nymph stage, and it'll feed a couple times on vertebrate host, and then it overwinters, molts, and finally into the adult stage here, where it takes the final blood season. So there's a lot happening here, and it's super complex, and it has this deeply embedded connection to the forces of wildlife and so surrounding all of these life cycles, what I'm trying to figure out is when you have common forest management practices, what is the effect on density and infect prevalence? You harvest timber on your property, cut down trees. How does that affect the density and infect prevalence? How does it affect animal security? When you fragment the landscape, you have forests over entire landscapes, start breaking it up. How does that affect ticks, better afraid stuff? These are relevant because this point is its life cycle. When it feeds on vertebrate host, that dynamic, those influence the success of that feeding, if it lives or not, and if that bacteria that causes Lyme is passed throughout the wild. Besides those biotic factors, we also have abiotic factors. Does anyone know what thing is? So Sarah's doing some real creepy with it. What is it? Yes, exactly. So how these little buggers do this is they climb up top of vegetation, put the legs out, they wait. They're like Velcro. So when the host walks by, they laugh. And that creepy little dance, that's their question. So majority of the time the tick is alive is actually spent in the It's either in the leaf litter on the forest floor, or they're being received crawlers and affecting on vegetation for a host. But then they get their host, they take a blood meal, and they're out. So that means that the majority of their life is actually off host. Those microclimate conditions, so temperature and moisture of the leaf litter, where that's like their refuge, where they go to moisture and all states, it can affect how long they live, their ability to mate, their production, full of the host community. They have the abiotic factors. And if we know we're in a forest system, which is where this tick lives, how does forest structure or characteristic what we use that forest, how does that affect the host, the questioning, the leaf litter? It's a very complex research. So what I went to do was collect various data to answer these questions I have. Forest, I'm asking about forest. I need to start collecting data on the forest structure and characteristics. I'm looking at the questing conditions and the litter conditions. I need to gather data on temperature, humidity. And for the host community, I'm looking at vertebrates. And there might be a multitude of whitetail, white vertebrates. It's here. There's also the involvement of predators of those animals. So it's also complex. And of course, no fun part. If I'm talking about this, I have to do the dirty work as well. So, first thing that I was looking at was, okay, we know that ticks are affected by the wildlife host. We know they're affected by temperature and humidity. And most likely, the structure of the forest is going to affect all of them, right? If you change the habitat of an animal, change the structure of the forest, it has all these effects. And it's going to affect both the animal and the fly out. So if we know this person, fix up. What tools can we use? What can we do? Film information to try to mitigate the problem. So that is where our very easy meta-analysis came from. That was published in. And what we looked at was forest management. How does changing the forest with these practices affect the wildlife, affect the microbiotic factors? Specifically, we looked at management practices that involve removal of invasive vegetation, prescribed burn, so applying a fire for relief vegetation, and then timber harvesting, going out on your own land, country, selling that, or using it. The thing here, so certain things that will be like administering prescribed burn. They'll be harvesting their timber on the property. So since it's happening anyway, and it's really common, it's essential that we know how to affect it. So, first question, what is the common forest and practice on abundance and infection problems? There's been a lot of studies in. So why am I also looking at the issue that even though there's all these studies, results from them are inconsistent. There's no synthesis. And what I mean by synthesis is how can I identify across all these papers, these studies that have been done, how can I identify a pattern and also quantify across all these studies? So that's where that systematic literature review is in, along with that. All right, so here, picture of the blood, sweat, and tears that goes into this. So this is from the book, The Handbook of Meta-Analysis, Ecology, and Evolution. So those droplets here, sweat, blood, take your pick, a little bit of laborious task, but it's worth it. But to start, you need to have questions or a pattern you know. We have this is our question. Then this whole area here is defined as scope, which I said is vegetation tools, and for harvesting. And then the fun part, real fun stuff. You have to actually search for that. And I'm trying to go out into the world and find every paper ever published on any of this quite a lot. So much in fact that when I search about science, PubMed, those research I use, I got 751 articles. I love to read. I love science. I don't think many of us want to read 751 articles very much really quickly. So I also figure out what's actually out. So reviewers, you have certain. Okay, here's an article. Let's check out the title and abstract. Does it obviously belong in this now study or not? What we found was that there was enough paper enough studies out there to look at the effect of invasive vegetation removal, dry burn. That's not enough to look at. 21 articles total, and there's only 21. Really not robust enough to now. So 751, 21. How did I get there? We have 751 efforts for all scientific searches. Then during the abstract revision, we realized, okay, there's 672 of these that just are not. So we're going to take this. Level 79, and those 79 were actually read and from the 79, we excluded additional ones because they didn't fit the parameters that we took list. So habitat type, if we were looking at own awesome. And we were left with 21 parts. How that breaks down is we had those 2 for timber harvesting, really on like shelf for qualitative literature review, and then we had 10 for invasive vegetation articles, and then 9 for stress. So, have my article. I pulled the data. I will not bore you. This is my data. My analysis. That's what I did. Okay. These are my results. So I ran 4 models to look at the effects of the Tick-Borne on density, the effect of Tick-Borne on infection prevalence, the effect of invasive vegetation removal on density, the effect of invasive vegetation removal on density. What I want you to focus on are those first 2 models, because those are the ones that prove. What's important with all those numbers is that the estimates for both are positive. Because they're positive, that means positive. Yay, there was an effect of these management practices on reducing death. And that p-value means that that's the thing. Love that. Love it. So this is what it actually looks like. You don't need to do that. Or yeah. So this was all the data that was pulled from the model that looked at the dry burn and their effect on tick death. And we found a negative effect, which means the dry burn may be useful tool for reducing. What I want you to focus on here, that little bitty done. You look at the bottom of the screen, see that number bar, it goes from -4 to +8. 0 means no effect. Nothing's really happening. One way or the other. Negative effect, not working, it's actually increased tick density. And then positive, being on the right side of the 0, means that it was effective, and this management practice didn't. So we saw that here for the dry burn, and then we saw it again here for the removal of invasive plants. So what all these numbers mean overall is that the removal of invasive plants, dry burn, timber harvesting, we looked at the studies out there, the effect was, we found timber harvesting, not enough, we need more. We don't have enough information to really run quantitative analysis, we just qualitatively report. But we did find that overall, it looks like the generalized whole trend is that remove invasive plants, reduce tick density. And the dry burn also be a substantial tool for reducing tick density. But please do not leave this room and think that, great, I hate ticks, burn it down. Don't do that. Don't do that. Prescribe burn, keyword. Also, there was a significant moderator. So the moderator means it's going to alter the effect in some way. So we know that prescribed burn is sensitive, but there was a moderator of time and what we actually saw is you do a burn, ticks go down. Awesome. We love it. 2 years later, ticks population rebound to be higher than what it was before the burn. So with the need, you have to be very careful with management. It can't be a one and done scenario. You might need to retry that burn, like you did, or at least around. Okay. So future direction from here. One big thing that was missing was timber harvesting. That's also my famous plug because when I did this, because we didn't have them, so I'm good. Then also looking at the long-term prescribed burn. Like I said, that was a moderator. So possibly having effective management is applying it over a long term, or we might just compare the burn to really what is the standard, like the flame height or duration. And then another area is that these studies looked at Ixodidae, so that infectious Lyme disease, they looked at amblyoma. There's another number of that were examined, but they really didn't compare to that. So some species are more robust than others. It's possible a burn successful in reducing Ixodidae population, but not. So So that's the thing. So that's we found meta-analysis. And like I said, we realized there needs to be more work done to carbon. So I was like, why not? Yeah, that's worth it. So that brought us to the second and third chapter. So we're looking at timber harvesting, and this is happening on the local spatial scale. Anyone give me an example of the local scale? What do I mean by local? Small town, yeah. So local means here, and in this case, with timber harvesting, this looks like maybe they have 1/10, or maybe they have 10. So there's a scale and a gradient, but it's happening that way. And timber harvesting is going to determine forest structure. Specifically, I looked at the number of trees per hectare as well as basal area. Basal area is kind of the way we form on the side. So the number of trees, how people. So the second chapter, I was looking at, okay, if timber harvesting happens on property, how does that alter the forest structure? How does it change the microclimate? We published this in the Journal of Medical Ecology. And here, I worked in southern Maine, so that's Portland right there. And I worked in a couple of different sites here. What I was trying to accomplish in site selection is what was in that middle graph. So there's a tree. On the x-axis, you have the number of trees per hectare. The y, you have the basal area, so kind of, again, size of trees. And you see that there's almost continuous range. And that's really helpful. So now I get the advantage. And on the third panel on the right there, that's kind of the whole serum. It's the block design, or finding my gradient. And within of these areas, I would have that was 70, and that's where I did it. Similarly, chapter 3, I'm looking at timber harvesting on properties, and now I'm looking at the mammal. I'm looking at these mammal communities, small mammals. I'm seeing what's happening. I mean, publicly. So similar approach to this project as well. I did a subset of those sites and I did a block design, and I looked at high intensity versus low intensity timber harvesting. So if I had a really high intensity harvesting timber, so I have a lot of trees. That took me up way more time than I'm. So if I did a high harvesting of timber, that means I have fewer trees remaining. A low harvesting means a lot of. So this is what the data collection looks like. I looked at the forest structure. So I call that my arrow compass, but it's a way to measure canopy closure. I looked at litter depth and cover, sapling count, and numerous other forestry measurements. I, of course, put my Ghostbuster suit back on, and I went to some good, fun stick dragging. And then I deployed the tool iBus. These little guys are really cool because they measure some personal information right out. And finally, I was looking at deer activity, and so for these large mammals, I took them. For the small mammals in chapter 3, I used what essentially is a metal box or Sherman trap, and that was allowing me to slide after small mammals like monks or mice. And I was looking at the population size, tick burden, or the number of ticks per animal, and then the infection prevalence. So is that animal infected with bacteria that cause Lyme? And then I also put out those super fancy foraging trays, which essentially is a plastic box that I can stand and there's a whole section side. And then to look at the overall activity, I deployed track clips, kind of a graphite mixture from a hard surface, and when the animal walks over, you get to the track. That's the way to measure overall. So, deep breath. I'm not going to go through this. Don't worry, I'm not going to. What I'm trying to show here is that the data that we collected allowed us to ask so many, look at so many different relations. We did a fun bunch of things, ANOVA models, other models, random forest models, that's ANOVA. Don't worry about it. You won't be quiz on it. I try to forget it too. But we did a lot, and it was really, really useful because it allowed us to answer a lot of questions. What we found, big relationship, we see here on this graph, where we have the density of Ixodidae nits on the y-axis and the number of trees per hectare on x-axis. And we see a very clear positive relation. So what we do is if we have a low number of trees protected. What do you think someone did on their phone? I've heard of, I believe what you're saying is that part of it, right? Yeah. So what this is showing is that forest management practice of harvesting timber make correlated lower extinction. So that's really important. Another thing of note, I'm talking about the size, black-legged thing. It is very small. Size for population. If you want, there's a photo online where it's a population comment. Someone has done the world a great service and put nips and sticks on there as well. And you can compare and see if you would be brave enough to try to keep that muffin without using it. Very difficult. Okay, so this is important because nymphs are the most epidemiologically life stage because they're the most likely life stage. So when I'm talking about ticks from here on out, I'm talking about the results of the found. So we found positive relationship and that's the model output. We have our p-value there. And we found that there was significant relationship between density of nymphs for CON, but not for NIP, is nymphal infection prevalence. So how many of those nymphs are infected for CIN, product of density. So what else we found was that timber harvesting, affects forest. So if you look at these two images, you see this one on the left versus the one on the right. The one on the right, way green, except all of that green is like hundreds of saplings. If I look at that picture, I see one tree here. These are really kind of saplings still, maybe. But over here, there's not much green, but you can count the number of large. So on the left here, you have a low harvesting intensity for trees. And on the right, you have a higher harvesting intensity for trees cut down, and for large trees, more sapling growth. And we saw that this was significant. So overall, with these numbers and these figures, what we need to look at is that both the number of trees per hectare and the size of these trees significantly affected the understory. So the vegetation cover provided by the understory, the number of saplings that are depth and cover. So we really have this large cascade effect down to the entire forest. The microclimate, which again refers to the leaf litter, temperature and humidity in that leaf litter, were also significantly affected, both the leaf litter and the testing. So both were significant. And what's the key, temperature and the humidity, bottom line, when you keep that overstory intact, when you don't harvest many trees, the canopy is intact, so not a lot of sunlight goes through. So things are relatively stable. So what we found was that when there were more trees per acre, had an intact canopy, pressed understory growth, but the microclimate also stabilized. The temperature and the humidity wasn't going all sorts of ways. Next, I used a random forest model, which is a statistical technique. It uses machine learning, multiple decision trees, but bottom line, what you need to know is it allowed me to assess out of all of these variables that I'm measuring, which ones are really driving the nymph density cover. And it gave us this output where it did a variable important all the things you're looking at. And what you can focus on are the top four. Now, think about forest, overstory, mid-story, understory. What are all of these here? Where are they? Down low. Yeah. So the most important variables that are driving the numbers of ticks that we see are really the understory. And that does make sense because that's what it's been for you. Also, questioning understory vegetation. So that was a forest structure. And then what we found from small mammals, that when we have a lot of, you have that intact forest canopy, you have a higher small mammal population. That was there. Found a similar pattern for their foraging. So small mammals foraged more when there were more. We found the opposite of this for some birds. And so the poor little guy here, if you look closely, you can see on his ears, got all those sticks. On side, one over there. actually count the number of things on that. And that's important. And so we found the opposite relationship. We actually found that when we had a higher for our density, but a higher level of burden. So that's a lot. Key takeaways. This is a relationship. It's showing us that harvesting maybe it's wool that correlates with lower density. Now, how does that happen? Why is that pattern there? For further research, so possible effect. So the core structure, if you have an infect canopy and it stabilizes the microclimate variables, it provides function habitat, that might be the mechanism, or it's the host species, and or it could be both. So the small mammals, there's population size, foraging behavior. So at this local scale, both backyard, we're going to harvest timber, possibly it could reduce the one. That's the local scale. Now everyone, big step up. Now we're going to the landscape scale. And at this spatial scale, which is much larger, I'm asking what's the effect of fragmentation or and on the predator prey. And so the landscape scale, we're talking square kilometers. We're not talking hectares or acres. So again, I was in southern Maine and I selected some experimental units along the fragmentation rate. So has anyone been here? A little bit? Okay. So Portland City. So you can even see here toward the city center, the fragmentation is much greater. Don't have as much forest. That out that urban area, it's less and less fragmented, more impacted. So we have this gradient, and it's kind of what it looks like from the here you have seen habitat, then the yellow that site that would have to like kind of this gradient have this site that's very fragmented, not a lot of habitat, and this is this one where when I say fragmentation, this case I'm talking about fragmentation density loss. What that means, I'm looking at it from all of the zones. I'm not just looking at total amount of habitat. I'm not looking at the number of habitat. I'm looking at things like in the entire landscape, what's the percent that's occupied by habitat, what's the mean size of that habitat patch, how isolated is that patch from its neighbor, the edge density, the edge of the habitat, how great. So really comprehensive and be exhaustive here when we're looking at all of the metrics, all of them to affect wildlife, their habitat. So if you want to know, you fragment the forest, what does it do to predators? So fox, bobcat, there's another predator in Maine and think of a wolf, coyote. Yeah, buffalo, fisher, very nice. So we're looking at these predators. We're seeing if okay, fragment their habitat, it affects the predators. Well, if you're changing the predators, it must mean something to their prey. So like small mammals, like put them out. And then if we change something about the prey that are common reservoir for Lyme disease and really important life stage, might affect it. So the link here might be presumptive. So if you fragment metrics, you have a fragmentation metric that fragments the landscape, it changes predator availability, predator activity, and that might change how many prey that predator is eating with respect. But there's another component. How else can we affect this besides non-specifically fear? So what I mean by fear is that it's not just big bad wolf going out and eating that. That now here's the that's going to hunt and fear and it's going to hide. And so it won't be able to vote much of the effort to important aspects forward. So if you have big bad wolves, in this case more like fox or coyote, the prey experiences fear, and the whole study of this, the ecology of fear, it's going to alter its behavior. And it might not devote much time foraging, and if it's not foraging around, it might not encounter things as readily and thus not support a higher. So all these things are connected. And one more component is deer. Deer are browsing species, so they often go to that edge, the edge habitat species, and so these fragmentation metrics might also affect deer in terms of So to look at this, again, data collection, we use trail cameras for large mammals. We use foraging trays to assess the foraging behavior of the prey. And as always, tried and true, we did some Tick-Borne. All right, one more big breath. So we looked at 16 fragmentation metrics total. And then we narrowed it down. Some of them correlated with one another. We were left with 7 fragmentation metrics that we really looked at. We ran a global model where we're putting all of these possible metrics, the model to see how it affects the density of ticks, simple, spectrum prevalence, density of infectious. And then we use the dredge function to be like, okay, these are possible variables. We don't know which one type is most influential. So let's dredge this, figure out all the combinations, and figure out what actually fits. And then from those top models, we ran the ones that performed best. So what this looks like is we had a model output where it's 128 combinations, but we are only looking at the top 4 because they fall delta kind of different out. Lower the AIP, better form. And then we ran those models, what's that? And that's how we determine how this is certain fragmentation metrics affect. And then put it all together. We wanted to see how predators, prey, ticks, fragmentation, how do they all relate? So on the x-axis here, we have the nearest neighbor distance, which means how close is the neighboring habitat? How close is my nearest? That's sensitive. On the y-axis, you have the nymph density, and you have the density of nymphs, density of effectiveness. What I want you to take from this, those are values or negative relations. So what we're seeing is that when the distance between neighboring patches of habitat increases, nymph density makes sense to think about it. Fixate small, not traveling. They're only traveling on the back of their host. And so if you have patches of habitat that are using and they're further and further apart, less of a chance of so it does make sense. Another method was that edge density. So when I'm talking about edge density, I'm talking about this part of the habitat. That interface habitat, the edge, and then the nature, which is the area. And again, it was significant. And we have that edge density on the x-axis here, the density is the y-axis. And here we see that it's positive. So when you have more of this edge, which often happens when you take a once cohesive forest, you fragment it and break it up, the edge density increases. So when you do that, you may also get your density. So that's the patterns we found that see fragmentation forest. Now, what about the wildlife? So what we see, which is a really fun cartoon, is all of the hypothesized beliefs. What we see with the dashed arrows here, so between deer activity and density of nymphs or coyote activity here, that's a hypothesized relationship, and it did not pan out. It means the data show that it was not. So we don't have data to support them. This very thick arrow here, what we're seeing is that we did have data for that relationship, and it's showing a positive relationship. So that means prey foraging. So when a mouse goes out and it's foraging more to focus on food resources, and it increases that behavior, it supports higher density. These other relationships that are shown here, but they're not dashed out, they have a couple of numbers. Long story short, is that one analysis didn't show it as necessarily significant, but it was a really labor-intensive study, so we only had 20 samples. We only had 20 studies, and a smaller sample size. So using another analysis, I'll bore you with it, is that those percentages you see in the circle, that's the probability that the relationship. So there's a 94.3 probability that edge density, that habitat density affects foraging behavior. There's a 87.7% chance that coyote activity negatively affects prey foraging. Same with the bottom, lower percentage, but it's still a negative relationship. So what this is saying, if you take one of the slopes here, is that increased predator activity, like fall, the new prey force fear, most likely. And then you look at prey foraging, it has a positive relationship with density of things. So if you have fox increased behavior, have increased prey foraging, and it's the same relationship with the tick block. So in this way, you have a positive relationship between foraging and tick, negative between predator and foraging. Ultimately, it's an that's negative, showing that increased predator activity can potentially. So again, that was a lot. Let me bring it all back together. So the local scale. So we're talking the whole backyard property they have, a couple of acres maybe. We saw through a meta-analysis that the dry fern and invasive maybe affected schools. We found that timber harvesting affects the forest structure, affects microclimate better, it affects small mammal communities, and ultimately also affects ticks. And then scaling up now to landscape scale, where we're talking square kilometer areas, we see that landscape fragmentation affects the correlation there between those metrics like density and near neighbor distance. And then we also found a cascading effect where predator activity alters prey foraging behavior, which in turn alters. And why this is important? It all relates to the ecology of land use. So there are going to be consequences for ticks and for ecology in changing forests. And we know with urbanization and human growth, that's going to be happening more and more. It also helps us inform management. I'm not saying that if you know these things about timber harvesting, that people are going to try to do it or not. They're going to do what they want to do. Like, however, form of that land. That's really powerful. That are prepared to that problem. And so we're hoping with all the research we can have stakeholders and the agencies, and so that could have done. And with that, have a good one.
Thanks, Steph. Anyone questions?
Thank you so much. That was a phenomenal presentation. So with a lot of the historical land management practices that we've done, there have been a lot of unintended consequences, and many of them to be, or at least many of the ones that we learn about in textbooks, be fairly negative. What would you want people to consider the potential negative consequences when implementing these land management practices, and what could be, maybe the drawbacks of having ticks or
Thank you for that question. So kind of twofold about like how about management practices, and side effects also actually I'll get to the last one. So the first one, kind of telling people that and practically and inherently on either good or bad. They are either focused. So I burn people will likely perform that dry burn, whether or not they have that management fine and they're probably giving that perform that dry burn. Oh, it might be safe. However, that now is made up of the so I think overall management is about so knowing that you might have a really great idea, you're going to use this tool to solve this problem, but it does not have about how that comes. And it's just what will we do with how that probably is more so goes off the like we don't know what we don't. Kids don't really but they are by birds, other animals. So they are other than that, can't really They all quite a point, but I always err on the side of often don't know what we don't know, and moving that might create a catastrophe.
There was another question back here.
During the period that you were performing this study, how many hours would you say a week were you out in the field?
That's a great question. Okay, so depends on which part of the study, which project, the meta-analysis. I am a physics biologist in that part, so the meta-analysis, 0 hours as a computer at that first matter, it's not great, but the performance, I still like it. For the other project, I'll give you the one that was the most, which is the landscape scale, where I was essentially hiking out miles of space with the ounce of sand in my bag to set up a foraging trade. Those days would be 10 hours? 10 hours? 10? I probably off that number. Now. Not 10 hours.
Thanks. I think I have imaginary ticks crawling all over me right now. So no,
Just to make sure everybody knows, we do not have large mammals on Appledore. We do not have endemic tick populations. So you saw that in the life cycle thing. So you don't need to be worrying about imaginary ticks on the island.
we actually have a lot. We have a lot of large mammals on the island, actually. Anyway, we'll do that. So yeah, you went through a bunch of land management practices and predator-prey relationships. I know those are different things. You're saying in some cases, for example, the tick population diminished. Is there like an order of magnitude there, like 10% or half or tenfold?
Oh, I won't try to look at it. That's a really great question. So we definitely published it where we were looking at the effect. So the magnitude of a reduction. And it's on that scale. So for harvesting, we found something like, okay, you have 100 square meters. You might reduce the tick by like 4 ticks. Doesn't really seem like a lot, but you divide it over a large area, and now it's the point of your 300. So that kind of becoming a personal question of like go back data and say, okay, for harvesting, maybe way, but how many? And is it worthwhile to use change with regulate? So probably not. Going back to that, probably won't change whether or not someone uses management, but if they're using it anyways, understanding what that next. Right.
Awesome talk. I have a question going back also to the meta-analysis and then some of the infection burden or tick burden work. I think it showed that like there wasn't a significant effect on infection prevalence of ferns and invasive species removal. And then it seemed like when you had lower mammal density, you had increased tick burden. I was wondering if you could talk a little bit more about kind of how these different, what the effect is overall on the small mammal infection prevalence and how that kind of maybe translates into transmission to other hosts of people.
So I would say mammal infection prevalence is always pretty messy too with all of it. So what we see here with the meta-analysis is was this tick infection prevalence. So in the 670s, they collected how many are infected. Small mammal infection prevalence. And I gave them really fun hearing whole bunch of my tested that tick. So here in the meta-analysis, the tick infection prevalence, we found no significant effect. Really what I found though is not a lot. And I think even myself, I tend to go, okay, how many ticks, how many am I seeing? I don't look at here, and less so about the infection level. This is part of the story. Going to mammal side, here we had the tick burden was off this, and kind of it does make sense with tick burden as least because what we see here where there are fewer small mammals, they had a higher burden, and essentially it's kind of showing that if you have fewer individual animals, then you can't spread the ticks out as you have dispersed across them, so they're more condensed on the tick. So that's the burden. For the mammal infection prevalence, we didn't see any difference. I honestly think it might be I think typically infection prevalence for tick population is like 30%. So select like 100 ticks, about 30 of them will be in or something. And because they're more dense, they're something and whatever goes by, there's a snag on. You're kind of running those odds across any mammal by any bird by. But it's a really good question because I don't think so. And more, we are more now, and there's like very bacterium and those better in certain than others. But it does come down to that. That small one really good at not safe.
Curious, when you looked at the landscape level ecology and you kind of zoomed out to the apex and mesopredators, was there an interest in taking that same approach when you were looking at land management? So how would something like timber extraction or burning affect some of those different
Oh, I think, yeah, so so sort of love to look at that, some of the competitors here on the local scale for stuff. So if someone were to harvest timber in their backyard, how does that alter predators? How does it affect that? Honestly, hadn't thought about it yet when I did this project, and so I hadn't got there yet. And also it's super labor intensive, so it's kind of outside the scope. But what we found here is we have increased foraging in the area where the vegetation happens. So we have like sunlight, but at night, moonlight. So I think what I'm thinking here, to your point, wasn't the carnivores, but I didn't remember 80 predators. What I like to think what's happening is we have a higher small mammal population that's foraging or still less appears here because of that impact over story, which prevents a lot of penetration. If you're not getting lit up with the spotlight, it's a lot harder to burn. I think that's a really good question. Thank you. And if I could go back in time to follow up, I would like to follow up.
Hi, thank you so much. Your talk was like awesome. It was really great. I guess I was wondering, you were talking about like sort of the prescription burn and the potential for rebound. And I was wondering about timescales and your other sort of factors. So were you seeing in land management for logging, like the various stages of rebounding in terms of like undergrowth, but then also like timescales for fragmentation? Like, did you ever see a potential impact on like this was fragmented yesterday versus 100 years ago, etc.? Thank you.
That's a really great question. So applying that, the time lag to all the different management practices, there's there. I think with prescribed burns, it's easier to document what happens over a number of years because it's a burn right away, it's gone, and then it is more like understory growth that goes faster. The super harvesting, you have that more catastrophic intervention first cut down the tree, but after that, hello, people didn't kind of do the same because trees took a long time. And so we didn't really work there, but where we were like, okay, if no one's put a tree down here in the last like 25 to 50 years, it's not harvested versus trees had to have been harvested years in the last 5 to 10 years. So 5 to 10 years was our scale. And for those we're looking at unharvested part. So the land fragmentation, that's also a tough one because hard historically how much it fragmented. Usually there was like with LiDAR, GIS now that but that it's another really interesting. We didn't do it in the study, but I think that how was most lifecycle overall.
Any other questions? We did not have questions online. Oh,
So this is definitely more of a foundational question, but where does Lyme's disease originate from and how have ticks, what's the story behind how it got?
That's a really good question. I don't have the answer. I don't think anyone does. It is kind of exciting. A lot of theories about it too. It's Maine floodline, Connecticut, and, you know, the government put it there. I don't, you know, don't quote me on that. So what makes it so difficult is that chicken and the egg scenario where we or an animal can only become infected with Lyme disease if an infected tick bites us, but that tick only got infected feeding on an already. So what came first, the chicken or the egg, the tick or the host? We're not really sure. Likely there was pathogen in a host that mutated the bacteria that now causes Lyme, a tick fed on that host, and that was how it started it. I think they traced it back to Europe. Possibly. Other than that, I really don't know. I don't think, I don't know if we ever will because it's kind of so complicated. So daring experience getting into that. So I don't know the cause, don't know where it actually started or its origin, and it does kind of give you a
Any other questions?
Hi, besides ticks, are there any other, like, I guess, ways that Lyme disease spreads that we should be aware of?
as of now, it is just ticks and also just ticks. So, if you ever are bitten by a tick, a lot of states have programs that send that tick in to get tested, they'll ID what it is. So it's great because you get bitten by a tick and you find out it's not there, there's not a huge. I've gotten asked before if you eat a tick, will you find it? Don't try it, please. I don't know. I don't think so. So right now, the only method of transient disease is infected tick biting us. So it should be fine. So anything else should be okay. But
Hello, that was super cool. I was just thinking about like, I understand like how do you test a tick, especially in its small like stages where it's still growing for Lyme? Because I was thinking like, yeah, you probably just like can sample blood from like creatures that are bigger, but ticks are so small. I'm just wondering how that works.
that is a phenomenal question. So once I have the tick, how do I tell if there's an bacteria in it? It's a laborious process, but essentially I take a vial, and then it goes through this whole PR process and essentially we find things. So either there's vibrated out override tick. I had to do a little more people. I for you people that but yeah, so this was a lot of time and really
Can Lyme disease spread only through ticks? And if so, why can't they spread through other herbivorous organisms?
question. Yeah, so they as of now, the only vector, which is the creature that carries the bacteria that causes Lyme, the only one we know of, and this specific bacteria. Why other ones? I'm not sure. A lot of times that kind of form where that vector is robust enough that carrying a pathogen won't kill the vector, but it's also not too robust where it's wipes out the pathogen. So it's able to carry it out of the side and then transmit it.
I want to thank Stephanie again for giving us such a important and interesting talk. It's always a little bit daunting when you hear something that you've heard so much about and then figuring out that it's even more complicated than you thought it was. For anybody who is interested, we do have our Rock Talks. We have 2 more for this season. Next week's Rock Talk will be Michael Coogan from UNH's Center for Sustainable Seafood Systems, who will be presenting Keeping Coastal Communities Alive from Fisheries to Aquaculture in New Hampshire. So if you're not on the island, you're welcome to tune in at 8 o'clock next week. And thanks again for being such a great audience, and thanks again to Steph for such a great talk.